Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we are moving on to the second part of the chapter number 7, that is Protists and Fungi, and I am your biology instructor Nushaba from Army Burnham College for Girls, Abbottabad. Topics of today's lecture include revision of life cycles of Physerum, that is the plasmodial slime mold, and OOMYCOTS, importance of protists to humans, general characteristics of fungi, and diversity among fungi. And today only two groups will be discussed, that is ZYGOMYCOTA and ASCOMYCOTA. Now first of all, the slime molds life cycle, which are fungi like protists. The group name is ZYGOMYCOTA. You can see in this amoeba like feeding plasmodium stage. This is called as plasmodium, although it is different from the malarial parasite plasmodium. This is huge amoeba-like organism having many nuclei, so a multinucleated mass, and it keeps on feeding, increasing in size, and then it matures to form sporangia. This stage is fungus-like, whereas plasmodium is amoeba-like. So two types of phases are found in the life cycle of slime molds. Sporangia produce spores by meiosis, and so these haploid spores may germinate to produce gamete-like amoeboid cells or flagellated cells which fuse in pairs to form diploid zygote. The process is called syngamy, that is the fusion of gametes. And this zygote, which is now diploid, undergoes mitotic divisions during which only nuclear division occurs and no cytoplasmic division, and it results in a multinucleated plasmodial stage again. So in this way the cycle repeats itself. Next is the water molds or OOMYCOTS. You can see filamentous structures having sporangia at their tips, and this is a potato tuber factored with the water mold. Some more figures showing mycelia. The water molds include water mold, white rust, and downy mildews. Downy mildews are the patches, brown patches on the leaf surface. Water molds are either parasites or saprotrophs, so they may be involved in decay or they may infect different plants. Cell wall contains cellulose and not chitin, so they are unlike fungi in that respect, and the feature is like plants. Filamentous hyphae, just like fungi are found. Hyphae are aseptate, mostly live in fresh water or salt water or in soil, so they are found in diverse type of habitats. Some are plant parasites, example is Phytophthora. Some animal parasites like those growing on the body of fish, like the one which is found in fish tanks known as fish fuzz. Motile spores called as zoospores are produced, which have two flagella. Zoospores are produced asexually in sporangia. Gametangia are produced for sexual reproduction, which are of two types: Anthridium, which is male, and Oogonium, which is female gametangium. Oospores are produced after the fusion of nuclei from male and female gametangium, and these oospores are the reason for naming of this group, that is OOMYCOTA. Here you can see the figure, the patches which are visible here in this video, which is self-explanatory. You can see the life cycle of Phytophthora infestans, how mycelium, which is made up of aseptate hyphae, grows in the intercellular spaces of leaves and green stems. The hyphae which grows into the intercellular spaces to suck nutrients from the cells are called haustoria. Here you can see a potato plant with underground tubers, branches. So here is the structure of leaf. You can see the mesophyll cells and intercellular spaces, the stoma, singular. Mycelium grows in the intercellular spaces when a zoospore falls on the thin layer of water moisture on the surface of epidermis, and this spore germinates through stoma into the intercellular spaces, where it sends out Hostoria, the parasitic roots, the parasitic hyphae, to suck the nutrients. In this way, this parasite damages the leaves. After some time, sporangiophores are given out through the stomata, and sporangia are produced, which contain spores. Spores may infect fresh leaves and fresh plants also. Cool, windy weather is very supportive for this parasite, and so motile spores are produced, which can move through the moisture to reach the fresh host cells on the leaves of the same plant or the new plant. They may also affect tubers. Now about sexual reproduction, as you can see in this figure in this slide, male and female gametangia are produced, the antheridium and oogonium. Antheridium contain male nuclei, oogonium contain female. Fertilization tube develops, due to which flow of cytoplasmic contents of antheridium into oogonium occurs. Then the fusion of male and female nuclei occur, resulting in the formation of zygote, which is diploid. Now this diploid nucleus then grows into zygote and an oospore. So this oospore is very thick-walled. It is dormant in the soil over winter. On return of favorable conditions, it may germinate and produce spores, flagellated spores through meiosis, which then infect the tubers and other parts of the plant. Now this is again the life cycle of OOMYCOTA. You can see asexual and sexual reproduction, as is explained in the previous video. Now about the importance of protists to humans. Now about the importance of protists to humans. Diatoms provide source, food and oxygen for the heterotrophs and aquatic ecosystem, as diatoms are the most abundant photosynthetic organisms in the oceans and the freshwater also, especially in oceans. Brown algae provide food to different animals, fish, birds, other marine organisms. They are also harvested for human food. And you know about kelps. They are also used as fertilizers. Red algae are economically more important, as agar is obtained from red algae, that is used to make drugs, capsules and vitamins. It is also used as a material for dental impression and as a base for cosmetics. In labs, especially microbiology labs, agar is used as a culture medium for bacteria. Now, disease-causing protists are another important aspect of protists. Plasmodium, that is the malarial parasite, and 1 to 2 million people die of malaria annually. That indicates how important this parasite is. Entamoeba histolytica causes amoebic dysentery. Trypanosoma causes sleeping sickness in man, especially in African countries. Phytophthora infestans is a plant parasite which causes late blight disease in potatoes, and will be discussed in the upcoming slides. Ecological importance shows that many protists in aquatic ecosystem form zooplanktons. Zooplanktons and phytoplanktons too, because phytoplanktons include algae and other photosynthetic organisms, and the zooplanktons include those floating organisms which are microscopic and they are animal-like. And so these protists are the primary consumers which form zooplanktons. They feed on phytoplanktons which are the producers. So zooplanktons are very important part of food chain. About Phytophthora infestans, it causes late blight disease of potatoes. It was responsible for the Irish potato famine of 1845 and 1847. 400,000 people starved to death because of this disease, because this parasite destroyed the huge crops of potatoes, and both leaves and potato tubers were infected. Next come the general characteristics of fungi. Fungi, they are studied under the heading of mycology, and the person who studies fungi is called mycologist. Habitat is aquatic, terrestrial, and as parasites on plants and animals. About their mode of life, they are parasites, saprotrophs, or mutualists. Parasites means they are dependent upon living host for their food and they may cause diseases in their host. Saprotrophs are decomposers which feed on dead organic matter, and they also destroy important materials. Mutualists are mutually beneficial organisms which form association with other organisms. Variation in size is also there. So fungi range from unicellular yeasts to large toadstools. Toadstools are mushroom-like. Mode of nutrition is heterotrophic and absorptive, lack chlorophyll, no photosynthesis, and in this feature they are different from plants. Digestion outside the body, that is external digestion, as there is an external enzyme system which helps in the digestion of food externally, and so direct absorption of nutrients occur from the outside. Cell wall contains chitin and not cellulose. This feature is also different from plants. Storage of food, if it occurs in the form of carbohydrates, is always as starch and never as glycogen and never in the form of starch. So this feature is also different from plants. Thallus or mycelium is the body of the fungus, and mycelium Greek word means fungus filaments. Mycelium is made up of hyphae. The Greek word hyphae means web, since hyphae form an intricate web-like structure. Non-motile fungi, all fungi are non-motile at all stages of their life cycle. They lack basal bodies, and you have studied about basal bodies in the chapter on cell structure and function, that basal bodies are found at the base of flagella and cilia, etc. Since they do not have flagella at any stage of their life cycle, so there are no basal bodies. And then they reproduce asexually as well as sexually. A side block information is given in your book that fungi are suggested to be evolved from red algae, and the reason is that both the organisms lack flagella in all stages of their life cycle. Here you can see the structure of hypha. Hyphae may be septate or aseptate. This is the mycelium composed of filamentous hyphae. Hyphae are tubular. They are made up of hard wall of chitin. Cross walls may be present or they may be multinucleated. And hyphae always grow at tips. In this figure, you can see the difference between non-septate or cenocytic hyphae and septate hyphae. Non-septate hyphae are those in which nuclei are embedded in the cytoplasm without a cell wall. Examples are ZYGOMYCOTA and OOMYCOTA. OOMYCOTA are water molds. They are not actually included in fungi. They are rather protists, whereas ZYGOMYCOTA are true fungi. Septate hyphae are found in BasidiomYcota and ASCOMYCOTA. These are also two groups of the kingdom fungi. In septate hyphae, you can see compartmentalization because of the presence of cross walls. Hyphae always grow from their tips because only the tip wall is plastic and it may stretch. Plastic means that it can stretch and then extend in size, and it is then retained, whereas elastic means something which regains its original position. So the remaining wall of the hyphae is rigid and it does not grow. Mycelia are ecologically active bodies of fungi. Now, fungi are considered different from plants in having no flagella, no chlorophyll, and they are saprotrophs and not autotrophs. Fungi are also different from animals in having a cell wall, although it is made up of chitin, absorptive mode of nutrition, whereas in true animals it is the ingestive mode of nutrition, and all fungi are non-motiles. And a unique feature among fungi is nuclear mitosis, means during cell division, spindle formation occurs inside the nucleus, so nuclear membrane is retained, it is not disintegrated. Being different from plants, animals, and the rest of the eukaryotes, the fungi have been assigned a separate kingdom. This is generalized life cycle of a fungus. You can see the asexual phase and a sexual phase. In asexual phase, mycelium produces spore producing structures called sporangia. Mycelium itself is it develops from haploid spores, so it is haploid, its nuclei are haploid. Spore producing structures sporangia are also haploid, and they produce spores by mitosis, which are again haploid, and on germination they produce mycelium. So this is the common mode of reproduction in most of the fungi, in which asexually spores are produced. In sexual reproduction, the hyphae of the two types of hyphae, they may fuse through their tips, and the flow of cytoplasm occurs between the tips that results in the fusion of cytoplasm called plasmogamy, and it may later on may be followed by the dikaryotic stage, in which nuclei they are present inside a single compartment in the form of pairs, but they are not fused. That's why they are shown here as n plus n and not 2n. This dikaryotic stage may be a lengthy one or maybe a short one, and it is followed by karyogamy. Karyogamy means fusion of nuclei. Karyon means nucleus. So the fusion of nuclei results in the diploid stage, zygote, and that undergoes then meiosis to produce haploid spores. These germinate to form the mycelium, which then may reproduce asexually again or sexually. Now about the diversity among fungi. Classification of fungi is primarily based on the type of their sexual reproductive structures and methods of the reproduction. The three groups are ZYGOMYCOTA, called as conjugating fungi, ASCOMYCOTA or sac fungi, and BasidiomYCOTA or club fungi. The naming of these three groups is based on their types of spores or spore producing bodies. Zygomycota are called so because zygospores are produced after the fusion of nuclei at their tips, and ascomycota are called as sac fungi because ascus is produced during sexual reproduction, which contains sexual spores. Basidiomycota are called so because their sporangium or fruiting bodies, that is club shaped, in which spores are produced. Now ZYGOMYCOTA, zygote fungi, you can see 600 species. They are cynocytic, that is multinucleated. Example is Rhizopus stolonifer. Black dead mold is the common example. You can see the spores, white and black spores are visible. Now strawberries having Rhizopus over them. Sexual reproduction occurs through zygosporangia. asexual reproduction is very common, and sporangia contains the spores. Sporangia are called bags of asexual spores. Hyphae have no cross walls, so they are xenocytic. They grow very rapidly over their substratum. They are mostly decomposers, pathogens, and some form mycorrhizal association with plants. Pathogens mean disease-causing organisms. Mycorrhizal association means the association between a fungus and the root of higher plants in order to increase the surface area for absorption of nutrients from soil. Since the tall plants, they need a large amount of water and nutrients from soil, and which cannot be done through the root hairs. So that's why mycorrhizal association provide the sufficient nutrients to them. This is the life cycle of Rhizopus stolonifer. You can see the bread growing, slice of bread having this mold growing on it. And three types of hyphae are visible: root-like rhizoids, stolon hyphae or stolonifer hyphae that is growing on the substratum in order to spread the mycelium, and vertical hyphae are sporangiophores, and sporangiophores bear sporangia or spore bags at their tips. These sporangia produce spores asexually through mitosis, and these spores then again germinate to form fresh mycelium over the substratum. This is the asexual phase. You can also see in this figure plus and minus symbols. These are just to indicate that two types of hyphae are found in the mycelium of Rhizopus, which are called as plus hyphae and minus hyphae. The two types of hyphae differ in some genetic characteristics in certain genes. So they are not called as male and female, rather they are known as plus and minus mating types. So the tips of the hyphae from two mating types, sometimes they come closer to each other and the tips fuse. The tips of the hyphae, they are separated from the rest of the hyphae through a cross wall, and then the flow of cytoplasm along with nuclei occur between the two, and this process is facilitated through hormones which are released from the tips of the hyphae. And here fusion of nuclei later on occurs, that is karyogamy, and it results in the formation of zygote, which is diploid. That diploid zygote then develops into a thick resistant structure having a very thick wall known as zygospore. That zygospore may remain dormant for a long time, or it may germinate in suitable conditions to produce spores through meiosis. Since zygospore itself is a diploid structure, but it produces spores through the meiosis. And so haploid spores again are released into the air, and on finding suitable conditions or substratum, they may again grow into a fresh mycelium. We will discuss ASCOMYCOTA in the next chapter, inshallah. So thank you for today's attention. Have a nice day, and Allah Hafiz.